we'll have more time to interact after this talk. Today, my name is Bjornsson. I've been in communication with some of you, and I'm the current director of the Epigenetics and Chromatin Clinic. You can see the logo in the middle there. And our idea with this clinic was to build a clinic that could develop the best expertise for a limited subset of disorders. And the disorders that we are focusing on, we call the Mendelian disorders of the epigenetic machinery. And one of those disorders is KAT6A. And I'm going to tell you a little bit about epigenetics. I'm going to tell you a little bit about our therapeutic development for Bougie Syndrome, which is a related disorder. And then I'm going to talk a little bit about what we could in the future hopefully do for KAT6A. Let's start with something very basic. So what is DNA? So DNA is the language of life. It's how the cells know what to do. And we sometimes call the entire content of the DNA the book of life, or the human genome. And if the human genome is a book, the genes would be words. So those are the kind of words in that book that make sense. And we've had this book now for about 15 years, but we still don't know how to read it yet. And the reason why this is very complicated is because this same text, this one book, the human genome, can be read in many, many different ways, at least 300 different ways. And the reason why I say that, there's 300 different cell types, and they all use the same book. And yet, some of these cells, like a sperm or a neuron, have very, very different components, so they must be reading the book in different ways. So how is that actually done? And one theoretical way that you can read a book in different ways is to highlight the part that you want. So theoretically, you could have a highlighting system that picks out the words that you need for a sperm, and then a different highlighter that picks out the words that you need for a neuron. But how is that actually done in cells? And I'd argue one way to think about the epigenetic machinery is to think about it as the genome's highlighting system. And so what are these? These are these, you heard a little bit about it both from Dr. Kelly and Dr. Harris. These epigenetic marks are these little tweaks to the DNA or the proteins that the DNA is wrapped around that are heritable through cell division. They're reversible, they're known to be affected by the environment or diet. And if nothing else, they add to the information content of the DNA. And there are two types. There's something called DNA methylation, and then there are the histone tail modifications. And KAT6A is a protein that affects the histone modifications. So in the histone machinery, there are four components. There are these writers. KAT6A is a writer. There's also erasers that compete with the writers. There's readers that keep track of what has been written or erased. And then there's something called remodelers that kind of move the nucleosomes around. So the way this works is that a gene is recognized by transcription factors, and there's a particular writer that's brought in. It puts on certain modifications. The writer and the eraser compete at that particular gene, and either that gene ends up being open or closed. And then you have these readers that can keep track of what has been done. So this is how, so the DNA is wrapped around these histone proteins, but the histone proteins have these tails that stick out of the nucleosome. So this is called the H3 tail, and this is called the H4 tail. And there's some very simple modifications, like histone acetylation, which is what KAT6A does, is a binary mark. You either have histone acetylation or you don't. And if you have histone acetylation, the chromatin is open and the genes are available. If you don't have histone acetylation, the chromatin is closed and the genes are not available. And this is the reason why we're so interested in KAT6A and these related disorders, because we think that the problem has to do with opening chromatin. That the genes that are needed are all there, but we just can't access them because the chromatin is closed. There's this other modification called histone methylation. It's a little bit more complicated because it has 4 states. You can have no methylation, mono, di, or trimethylation. This is an open chromatin mark, H3K for a trimethylation, but then there are other histone methylation marks that are only seen in closed chromatin. So what I want you to get out of this is the histone machinery consists of writers, like KAT6A, erasers, readers, and remodelers. And there are many different histone modifications, but what I want you to know is that there are some marks that are only seen in open chromatin, like the acetylation, and then there are other combinations that are seen in closed chromatin. So what then happens when the epigenetic modifications or the machinery are disrupted? So this is the basis of our clinic. The idea with this clinic, which is myself, Jackie, and Jill work in this clinic, is that we could build expertise in these disorders and be ready for therapeutic trials, develop biomarkers, and also just build expertise so that we can be the best providers possible for the patients with these disorders. And we see a lot of patients with classic epigenetic disorders, but we also see a lot of patients that have defects either of the DNA methylation machinery or the histone machinery. And there's now about 55 of these disorders. I know this is a complicated figure, but I just want to walk you through it. That when we wrote this review, there was about 18 writers, 7 erasers, 6 readers, and 13 remodelers. And the other thing that you can see when you look at this wheel here is that almost all of them have this yellow label. So this yellow label is when the kids have intellectual disability. So basically all these Mendelian disorders of the epigenetic machinery lead to neurological dysfunction. That's a unifying feature of this entire group. There's other features. For instance, if you look at the red label, that means the kids have growth retardation, so they don't grow as well as other kids. And almost all the disorders have either growth retardation, and then there's a small subset that has overgrowth, so the kids are too big. So intellect and growth seems to be severely affected by these of these disorders. And then the other thing is, these disorders are enzyme disorders. So KAT6A is an enzyme. And usually you need to lose both copies of an enzyme to get a disease. But in these disorders, you only need to lose one copy to get a disease. So it's enough to lose one copy of your KAT6A and you end up with a disease. So that's another unifying feature of this entire group. They're very sensitive to the amount of the factors that are needed. And we now know a lot of these writers and erasers that are associated with the very marks that I showed you that lead to disease. And you can see, we see a lot of patients with something called Rubinstein-Tabby in our clinic. And KAT6A has overlapping function. And it's very interesting to listen to you talk and tell us about what your kids have, because we see a lot of overlap between Rubinstein-Tabby and KAT6A deficiency. Rubinstein-Tabby has similar intellectual problems. They also have very severe constipation, and they also have growth abnormalities. So there's a lot of overlap because we think that at the end of the day, maybe some of the downstream targets are shared between the different conditions. So here I'm just putting some pictures. I haven't added a picture to KAT6A because it's so new, but basically it maintains some of the acetyl marks here, just like the Rubinstein-Tabby factors. And many of the patients have some phenotypic features, but I think the other thing that's someone asked before about rare birth defects. So these conditions affect many, many genes because they regulate many, many genes. And I think that in general, the phenotype is quite broad in these disorders. There's a lot of different disease things that can go with a condition because of the fact that this is a transacting factor that interacts with a lot of other genes. So maybe depending on what variation you have in those genes, that's going to decide how your child ends up having, what feature your child ends up having. So to summarize here, the Mendelian disorders of the epigenetic machinery, like KAT6A, these are genetic disorders that disrupt the epigenetic marks. And they have very they're very dosage sensitive, and that indicates that the marks are very tightly regulated. And we know now of many conditions that are caused by mutation in these writers and erasers, including Bougie Syndrome and KAT6A deficiency. So the question that we always get, and you know, the dogma in genetics has been for a long time that you cannot treat intellectual disability after birth, that all the damage has already occurred and there's nothing you can do. However, we have some evidence, and I want to show you the evidence, that there is some flexibility in these disorders. We treat them after birth. We just have to figure out exactly how to tailor that treatment to the individual conditions. So, and because I have most data from this disorder, called Bougie Syndrome, I just want to show you the data so you can understand what can be done in the future for KAT6A. So Bougie Syndrome is a disorder where there are two different genes known. The kids are all small, the kids all have some intellectual disability, and many of the kids have immune problems. And so neurologically, they have intellectual disability, and they also have very bad visual spatial defects. So what that means is their drawing is worse than a lot of other things. So they basically are very poor drawers. It also affects how they interact and learn in their home. So they often have great difficulty kind of figuring out where they're going and so forth. And there's some overlap with KAT6A there. Kabuki often has hearing loss, which is not something that we see in KAT6A at this point. They often have congenital heart disease, which we do see in KAT6A. They do have some immune problems, endocrine problems, growth problems, skeletal and renal problems. So, the reason why I was so interested in Bougie Syndrome is that there are two causes. There's a writer and there's an eraser. So the writer that's defective in Bougie Syndrome is called KNT2B, and it puts on an open chromatin mark that helps keep the genes accessible. The second cause that was found is this eraser that removes a closed chromatin mark, and again, even though it's a different mark, they both have in common the inability of opening chromatin, just like in KAT6A. So my idea, which was a very simplistic idea, but this was our starting point, that maybe the balance between open and closed chromatin is very tightly maintained, and maybe in Kabuki and KAT6A, the balance is tilted towards closed chromatin, and if that was true, we should be able to use drugs that open chromatin and get some therapeutic effect. So to do that, I developed a mouse model for Bougie Syndrome, where we knocked out the enzymatic function of KMT2D, and we were able to show that the mice had many phenotypes that we see in the patients. They're small, they have skeletal abnormalities. This is work from Jill, who you guys can talk to later. She's been very focused on the growth in many of these disorders. And because we were interested in intellectual function, we did a wide variety of behavioral tests in these mice to look how they compare to other mice that don't have these mutations. And this is an assay called novel object recognition. So here you put identical objects into a cage, and then the next day you put a new object. And a normal mouse should spend more time with a new object because they remember already seeing the other object, and mice are curious. But when we did this in our Kabuki model, what we saw is that there was no difference on the first day, but on the second day, the mice they spent less time with the new object, and that suggested that there was a problem with hippocampal memory. So we did another test called the Morris water maze. It's a tank filled with water. The water is made opaque with a dye, and then we teach the mice to find an area that has an invisible platform. And we train the mice for 5 days. And over the 5 days, the mice get really good at finding that platform. And then the last day, we get rid of the platform and count how often the mice go through the area where the platform used to be. And when we do that, what we find is the Kabuki mice cross about half as often as do the wild-type mice. So again, there's a problem with hippocampal memory in these patients, in these mice. We also find that there's a biochemical abnormality in the Kabuki mice. When we compare wild-type mice to the mutant mice, there's much less of this mark, H3K for trimethylation, throughout the genome in the Kabuki mice compared to the wild-type mice. And you can see that there's this skewing away from the average. However, if we put the mice on a drug we can rescue this biochemical abnormality so that there's no longer any difference between the wild-type mice and the kabuki mice. So that was encouraging. So a summary here is there are two types of Bougie syndrome that suggest that the balance of opening chromatin has something to do with the phenotype. And our mice have many features that are similar to the patients with Bougie syndrome, but they also have hippocampal memory defects, which is something that we can keep track of during therapeutic trials. And finally, the mice have a defect in putting on that epigenetic mark, and we can affect it with drugs. So the next thing that we did is we wanted to find if there was a cell population that we could keep track of during therapeutic trials. And what we found is that there was a lot of this factor, KMT2D, in this area of the brain called the granular cell layer of the dentate gyrus. Now this is a very cool area because it's one of the few places where we have newly formed neurons being formed throughout the life of individuals. So this is the place, so most of the neurons are made in the belly when you're in the womb, but there are a couple of places that keep making neurons throughout your life, even into old age. And it was known that if you disrupt this process, you get hippocampal memory defects, which is what we see in our kabuki mice. So we next wanted to see whether there was actually a deficiency of making new neurons. And the answer to that is yes. When we look at the wild-type mice, which are shown here on the left, we see a lot of these double coordinate positive cells, but when we looked at the kabuki mice, we saw many fewer of these green double coordinate positive cells. And when we quantified this, we found that there was a massive deficiency in the kabuki mice compared to the wild-type mice. So then the question was, can we actually manipulate this? Can we, even in postnatal life, can we treat that phenotype? So here we've treated a full cohort of mice with a drug called AR42, which is an HDAC inhibitor. And the wild types are on top, the mutants are below, and you can see that off the drug, there's a very big difference. Many fewer double coordinate positive cells, less H3K for traumethylation. But as we increase the dose of the drug, the mutants start looking like the wild type mice. So you now have a rescue where now we can see these cells where we couldn't see them before. And when we quantify this, we see that there's a dose-dependent effect. As you give more of the drug, the mutants catch up with the wild-type animals. And this worked both in very young mice, 1 to 2 month old, which is like a young adult in patients. And it also works in older mice, 5 month old, which is like middle age in people. So then the question was, can we actually affect the functional output? Can we make these mice perform better in memory tasks? And remember, we had this defect where the kabuki mice shown in yellow did much worse than the wild-type mice in blue. On the drug, they both did better, but now the kabuki mice were no worse than the wild-type mice. So yes, we seem to be able to treat the functional effect as well. So the summary here is a mouse model of Kabuki syndrome has impaired neurogenesis in the granular cell layer of the dentate gyrus, and defects in the dentate gyrus can be reversed using drugs that target the epigenetic deficiency, and that suggests that the intellectual disability in Kabuki syndrome and other related disorders like KAT6A may be treatable in postnatal life. So this is all good and well, but not everything that works in mice works in humans, right? And the problem is we don't have brains from kids with KAT6A because most people don't want to give a brain sample. So what can we then do? How can we study this in our patients? So in Kabuki syndrome, if you have defects of adult neurogenesis, you would expect very bad visual spatial effects. So Jackie has gone ahead and tested about 25 patients with molecularly confirmed Kabuki syndrome. And what she finds is that these patients have very severe visual spatial effects, which is shown here in blue, but they have much better other functions that we can measure, shown in red and green. So again, this kind of supports the idea that we are barking up the right tree. We're looking at the right area of the brain. The final thing is the other type of ways that we can study this is we can try to define a biomarker, something that we can measure in blood, and we can keep track of as we treat patients in a clinical trial. And what we found is that a lot of these disorders, including Kabuki syndrome, have defects that is very easy to measure in the blood. Defects of another system called DNA methylation, so it's another epigenetic mark. And we have found sites in the genome where which differentiate the kids that have Kabuki completely from normal controls. So we are hopeful that this will act as a biomarker and that similar studies could be done for disorders like KAT6A. So just to summarize here, the patients with molecularly confirmed Bougie Syndrome have visual spatial defects, symptoms that localize to the dentate gyres of the hippocampus, and patients with molecularly confirmed Bougie Syndrome have DNA methylation abnormalities that may act as a potential either diagnostic or therapeutic biomarker. So what are the opportunities for KAT6A? So it's very interesting that we would argue, and this is something that I argued in a publication with Jill, is that maybe the problem in these disorders is just a balance between open and closed chromatin. And if that's the case, some of the treatments that have been determined in other conditions may work for KAT6A. And by doing something like an HDAC inhibitor or something that works like an HDAC inhibitor, you might get some therapeutic benefit. And that's why I was so excited, because carnitine kind of works like an HDAC inhibitor. It potentiates open chromatin. So theoretically, from an epigenetic standpoint, the carnitine treatment makes perfect sense. It makes sense that you're seeing response to that. And it also suggests that there may be some therapeutic flexibility in KAT6A. And it means that maybe we could, over time, develop other agents that are even more potent than carnitine that could help treat patients with KAT6A deficiency. But I'm an advocate for preclinical studies. We need to publish some of these results. It would be nice to publish the comparison of kids on carnitine versus kids that are not on carnitine. And it would be very nice to do mouse studies and see whether we can figure out what cocktail or what medications are most efficient in a mouse model of this disorder. So, and then the final slide that I have, I just want to say, how can we work together? So please consider participating in research. It doesn't have to be with us, it can be with anyone. Because I think the more people that take on the KAT6A cause, the better off you're going to be, the faster the science is going to go forward. Consider fundraising, because you should fundraise for your foundation, and some of it should go back to your patients, but it also is very powerful when family groups can kind of seed research. If you can give a researcher a small amount of money, sometimes that's enough for that researcher to get preliminary data and get a bigger grant. And now that person is on the KAT6A path, so you can cultivate researchers by fundraising and providing small amounts of grant money. Organize meetings. I'm very proud of Jackie to organize this meeting. I think this is the way you start progress. We hear from you what you want. We can tell you what's possible and how it should be done, and then it allows us to kind of move forward. You should also lobby for this condition. If you have connections, the problem that KAT6A and some of these other disorders have is that they're so rare that no one cares. People have never heard of this. The doctors haven't heard about it. You can just imagine how the funding agencies act when we try to get funding, because they're like, what? KAT6A, I've never heard of that. So lobbying and pushing and getting the name out there is very, very helpful to move the science forward. And we've done many such things for Kabuki, Rubinstein, KB, and we feel like that it has made a big difference for these disorders. Okay, so this is just the people that I'm privileged to work with. You can see both Jackie and Jill. I encourage you to consider coming to our clinic. Like you heard this morning, I have many roles. I work both here and in Europe, so it's a little bit hard to get appointments with me, but these people have great access to me and work closely with me. And I think our clinic, the Epigenetics and Chromatography Clinic, would be a perfect home for patients and families of kids with KAT6A deficiency. So I'd be happy to take any questions. Thank you so much.
I actually have a question for you. So, being in the military, obviously I have to get referrals to potentially come see you. what is the likely of, like, how would I be able to do that?
So we see many people from the military,
Okay.
but you need to get like approval from your geneticist within the military.
Okay.
And you know, so one question is, what's the benefit of going to a center that sees a lot of kids with? Is it worth your time?
Yeah.
And I think it depends on where you live.
Yeah.
You know, genetic services are very unequally distributed. So in the East Coast, there's a lot of genetic great centers that are probably decent. The middle of the country, not so much. West Coast, again, a lot of good geneticists. So it depends a little bit on where you live, and I think each patient should decide for themselves.
Okay.
Do you feel like you've got suboptimal services? Then it may be worthwhile making that trip here. We try to have our clinic on Fridays because that's very convenient for families that are traveling in. Let's say you want to go to DC anyway, you could come see us on a Friday and then go to DC for the weekend. So, and what would we do at such a visit? So we would look over, make sure that all the studies that need to be done have been done. We would make sure that, you know, the carnitine was appropriately dosed and so forth. And we would make sure there are not other studies that your child needs. For instance, the literature, because it's such a new disorder, there's constant new articles. You know, a very busy genetic service may not have time to look at that, but we are very interested in these conditions. So we look at every single paper that comes out and we think about it. For instance, for KAT6A, there's now been described a couple of kids that, in the Netherlands that have deficiencies of hormones, and that can affect their growth. So that's something that our clinic is all over. We want to see if we can find kids that have that same problem. So I think that's the benefit of going not only to us, but to a center that wants to, you know, see kids with KAT6A and help build expertise. Other questions? Yes?
Yeah, concerning the hormones, you know, our kids are little. Most of us here have little kids, 10 or younger.
The clue in that paper is that this is a paper by Dr. Hennekamp. It's a couple months old, or 3 months old or something. But the clue is the ratio between the linear growth and the weight is abnormal. So what happened in that patient is that they stopped, the height stopped, but the weight kept going. So if your kids, if the pediatrician says, oh, I don't understand why the height has stopped growing, but the weight keeps going up, those are the kids that need to have that hormone testing.
My son, it happened this year. He was always on the height going up. Now he's stable. I noticed because his brother is 16 months younger and he was always taller than his brother. Now he's wearing clothes like that just don't fit even his brothers. And his height stayed stable over a year now. He's not growing taller. That's why I'm thinking about it.
So, you know, I think anytime you have a child that does that, where the height stops and the weight keeps going, you want to screen some of those hormones out.
And you do this in your clinic?
Absolutely.
Because I went to an endocrinologist in my area, everything is normal, so you stand up, you know. But I don't feel it's normal because he was always growing on the curve. And
Did they check any hormone levels before they told you it was normal?
PSA, very cute.
But you know, so in the child that Dr. Hennekamp described, there were problems kind of centrally in the brain.
Yeah.
So it was not downstream hormones, but it was like the upstream.
Must be triggered. I read that.
Yeah, exactly. That's exactly what it was. So what I'm saying is, you, as a foundation, you should pick a couple sites or 3 or 4 sites, because not everyone can go to California, not everyone can come to the East Coast, but it's nice to have advocates for KAT6A that are distributed throughout the U.S.
We're trying to do that in the U.S. The bigger problem is not the U.S., is Europe and other countries. But we, you know, Australia now, we're trying with Australia. Europe is still a challenge. In the U.S. we have you here, and in Maine we have Dr. Smith, Wendy Smith, who's willing to see KAT6A patients. Not sure if you know Dr. Wendy Smith. In California we have Dr. Adelaide, that's only a researcher, doesn't see clinical patients, but we're trying to have.
So I can see patients in Europe through Iceland, so that's another possibility.
Great. Because we have family now, one family in Austria, a couple I think in Germany, too many in England,
Yeah.
One in Belgium, they don't even do the whole exome sequencing. The family has to go to Paris to get the blood work.
So it's the same in Europe as here, is that there's a limited numbers of centers of excellence that are going to be uptaken. But I think, you know, today, your biggest strength is that you're connected via social media. So you can quickly figure out where the good centers are that are useful to you. And then, you know, you go to the places that are most useful to you. Yes?
Related to the hormone question, Jack has had his growth hormones checked and they were told that they were normal and they weren't sure if he would benefit from the injections or not. They said that we could try, but that it could contribute to central apnea.
Yeah, so I think that's a really good point. So for kids, so growth hormone has been tried in many different conditions in this group. And it has, overall, the jury is still out whether it's beneficial or not. The thing that I want to say, any child that has hypotonia, low muscle tone, needs to have a sleep study before starting growth hormone because there is a risk, not so much with central apnea, but that the tonsils and adenoids can grow, and that if you have mild sleep apnea, it could get worse, and that's another thing that can affect your development. And we want to give kids with KAT6A deficiency the best chance to learn and function. And we don't want them to have something that's treatable like sleep apnea.
He is on a machine. He's had tonsils, adenoids, basic tongue reduction surgery, which got rid of the obstructive, and then he's had decompressions, tracheary for the central apnea. So, but he still requires, he has chronic respiratory failure and retained CO2.
The problem with the growth hormone is that it is not, so KAT6A is not a clinical indication for growth hormone. So most endocrinologists would have to have other data to support it for your insurance to cover it. And growth hormone is like crazy expensive.
Yeah.
If it's not covered, you're probably not going to do it.
They benefit of headaches constantly increasing and apnea getting worse, so not doing it at all. Is it a big deal that he's short?
But this is the reason why it's so important to get the baseline. And one of the things that Jackie has worked on, not only for Kabuki, but for some other disorders too, like rheumatoid TB, is she is figuring out how to measure the baseline function. Because if you have the baseline, now you can start doing clinical trials and see if you can improve the function. But without having those basic data, it's impossible to know, like how are you going to know if things are going to get better or not? You have to have some kind of metric to measure it.
Yes.
So my work has mainly been on mouse work, and I think there's room for all kinds of talents. I mainly do mouse work. Jackie and Jill have done more of the clinical stuff. Jackie is doing like MRIs and testing to test the visual-spatial function. I think those are studies that would be very helpful to have.
Is it around 35 to 40 per mouse? Yeah, the cost per mouse was about 35 a few years ago.
How much does a mouse cost?
Yeah.
It depends on what kind of mouse it is and what you're going to do with it. So the mice
Not feed it to a snake.
So the mice are very, very expensive, and it takes time to do the studies. Like the stuff that I described, I've had a grant that's about $250,000 per year for 4 years. So that work took maybe 3.5, 4 years, $250,000 a year. It's very expensive. But you may not, as a foundation, you don't start out by fundraising a ton of money. It's also very helpful to have small amounts of money. The first grant that I applied for was $10,000, and I got a little bit of data from that. And then I got a bigger grant, and that's kind of how it works. But sometimes a little bit of money can go a far way. Yes?
So with KAT6A, my understanding is that when it's underexpressed, you get this disorder. When it's overexpressed, you can get myelocellular leukemia, or it can promote leukemia, right? So my understanding is there's a lot of mouse models that have been developed for KAT6A associated with the cancer-focused research. Is it the case that those mouse models could be leveraged for this work? Or
I promise you that there are probably multiple models that have knocked out KAT6A, because in the scientific world, there are many programs that are systematically knocking out genes. So the limiting factor is it's expensive to buy those models.
Even when they exist. So it's the cost of creating them, it's buying access to use.
Yeah, so it's
Okay.
usually like $5,000 to $10,000 to get a mouse. So because most of the time the way the mouse is kept is not living. But what they do is they take germ cells, sperms or embryos, they freeze them down, and then you order them frozen. So that now the people have to do IVF with that. So the total cost of buying that mouse is something like $5,000 to $10,000. You have to ship it here, it's in quarantine, maybe you need to back cross it to make sure it's on the right genetic background. So, so that's the limiting factor. It's usually not making the mouse itself. Yes?
So I'm not sure if you're aware about the Australian mouse, Dr. Boss Center, because we have been in touch with her, and she's willing to work actually with Dr. Kelly and with you. She has a mice.
I think that would be wonderful. I think that would be wonderful. Like I said, usually there are plenty of mice available. It's more that, so if it's very hard for people to buy into something because they have to maybe pay for the salary of the person doing the work. It's easiest that people have like a chunk of money they can dedicate to this. That makes it easier for us to move the work. It doesn't mean that we're not trying to move the work, and we can move the field in the clinic, in the beginning, and then once we collected clinical data, maybe we can use that to propose something. But like I said, for a lot of these rare disorders like KAT6A, the funding agencies have no clue what this is, and because it's not common and there's no pressure on them, they're less likely to fund something that they don't know compared to something that they know. So that's one of the problems that we kind of we're up against. Yes.
Going back to the growth moment, you mentioned that it's not growing tall while gaining weight, it's progressive, it's not the other way around as well. Like my daughter is growing tall, but her weight is, so she's 5.5, she's 36.
It's usually less of an issue. We're less, like if the child is very, especially if the child looks proportional, I think it's okay.
If there's sort of a reason, right? Like if she doesn't eat a lot, I mean,
But is she active? Like,
yes, yeah.
some kids that are very, very active just burn a lot. Yeah, burn a lot of calories. So often you see once you go from crawling to walking, the weight kind of takes a hit, and then you reach equilibrium later. But I think usually that's less of a concern. Yes.
Talk about providing some small grants, like research and and then would you be willing to give advice to the foundation about
Absolutely. The way to do this is that you create a scientific board for your foundation. It's people that are knowledgeable and willing to give their time, because usually you don't pay those people. And then you fundraise, and then you advertise, and you try to get as many people to apply as possible. And the reason for that is sometimes the people are just thinking about it, and they own that grant, it makes them more likely to reuse that language for something else. So you want to kind of create a buzz about KAT6A, have a bunch of people apply, and then you want to have a way, usually a two-tiered way to prioritize the grants. The first should be scientific review by a specialist, where that person says, these grants are okay from a scientific standpoint, and these are not. Because there are sometimes grants that are just a little flaky. After that, then the families should decide who among the acceptable ones is closest to what you want to achieve. And it varies from patient group to patient group, what people want studied. In Kabuki, everyone said, we want intellectual function and anxiety. Those are the things that we think need to be studied. When we go to Arubis and Tabi, they say, oh, we'd like behavior and constipation studies, because those are our biggest issues. You as a community should decide for yourself. You write a grant, it's called an RFA, a proposal, and then people can submit to that and try to mirror what you want to get out of it.
would you be willing to participate on that?
Absolutely, absolutely.
Sorry, just to follow on that. So we are going to move out as the foundation on creating a patient registry. I think we are at this point planning to use the registry platform provided by Nord. for rare disease?
The other, there are, NORD is one, and then there's another one called Sanford. Sanford has free registry collecting tools, so some of the other foundations work with them.
Okay. Regardless of which platform we go with, right? We've got a sample. Would you guys be willing to consult on the list of questions, the survey that we put together for the registry? Right? We've got a good, we're with Dr. Arboleda because she put out this survey that is the genesis of the paper that's going to be coming out, just characterizing the patient group. But obviously, tailoring the registry questions is vital and important to collecting good information for eventual trials. So,
and it's going to change.
Yeah, it'll change over time, but we want that initial set to be as well known as possible. So, if you guys are willing, obviously, we'd love support in reviewing the questions we come up with to validate that yes, those would be effective registry questions.
Absolutely, we'd be happy to.
And then another question on, I think, my understanding is that when you're going into trials with the FDA forever, and maybe still is the case, right? You want the measures of your study to be on actual human impact and performance, right? But I think with the recent approval of this treatment for Duchenne muscular dystrophy, I think it was like the first time where they approved actual measure of a biomarker level as opposed to demonstrated change in patient behavior, right? So is it the case, you expect that, as you highlighted with the Bougie and whatnot, if in KAT6A, if it's hard to pin down one aspect of patient performance to measure in a drug trial, do you think it will be likely that we can successfully use one of these biomarker levels as opposed to...
So,
Got him.
When you're designing a clinical trial, the ideal, the perfect thing is actually to have both, right? To have a biomarker, which is something that you can measure and quantify, right? And also to have some functional studies, right? The issue is that Duchenne muscular dystrophy is very different from KAT6A in this particular question for a lot of reasons. Number one, it's a much more common disorder. And number two, it's a much more well-known and well-studied disorder. So those functional performances that you're talking about, they're very known, they're very there, and they're very easy. So a trial could get approved by saying, yeah, we're going to do it with this biomarker, right? This is the new part about our trial, is this biomarker. But we're just going to use the same functional studies that all 500 trials of Duchenne muscular dystrophy has used before us. For KAT6A, we don't have that information. We don't know what functionally we need to measure. And we don't know that on a lot of different levels. We don't know that on what is going to be important to you guys to measure, what is going to best affect the quality of life of your children. And we don't know that on what are the best measures to use to test those things that turn out to be important to you. And that's why doing it in the way that we try to do it here is really important to us. And sometimes even like foundations and parents get upset because they want things to move quicker, but it's really important to us that we do the preclinical work, which means the mouse data and also just sort of getting the information like from registries and things that you're talking about. And then we develop outcome measures clinically, clinical outcome measures for the things that matter to your kids, and test those and then go into a clinical trial ready, okay? Because what sometimes can happen, and it just recently happened in a disease called Fragile X syndrome, which is another genetic cause of intellectual disability. They did all the good preclinical work, and they had a drug that they thought would help, okay? But what they didn't do very well is that middle step, that getting good clinical outcome measure data. And so they did this huge trial and tested the drug, and they did not see the results that they expected. But what happened was, at the end of the day, they're still not sure Did they not see the results they expected because the drug really doesn't help Fragile X? Or did they not see the results they expected because they didn't look for the things that they were supposed to look at to see if it helps Fragile X? And so that is, that we just feel that that's really, really important. And we definitely try to move those things as quickly as possible. But we feel that, even though there's an eagerness to move things along, that if you don't take that extra year or two to get that data, then you may set your disease back, especially for something super rare, 10 years in getting down the therapeutic pipeline.
Yeah, and I think that's what, as a group, we'll need help with. Say this HDAC inhibitor looks like it might benefit a bunch of different chromatin modifying disorders, right? Wow, like, important for us to understand, right? That could still be years away from us being effectively able to try it, and there's all that organizational work we need to do in advance of that. And so,
but that's why it's so important for you guys to sort of think about that, the early sort of stuff, and what you as a foundation want to look at and fund, so that that work can be ongoing while, say, HDAC, any sort of these trials are being tested in Kabuki or Rubinstein TV or anything that KAT6A may want to piggyback on, so that you could say, okay, well, it worked in these other things. We also have measures ready for KAT6A, so now we're jumping on board. And that's how you get things done quickly and efficiently for rare diseases. Because unfortunately, there aren't drug companies clamoring at your door to treat just KAT6A because it's so rare. But there will be if it gets grouped in with a lot of other diseases.
Yes?
So in terms of data collection, is it important that we bring our kids to see you and also to see Jackie to set up those rules?
So the way we usually work in the Epic, I know it wasn't the case this week just because of the situation, but usually we try to pair up with Jackie. So the way we built this clinic is Jackie and a geneticist sees most of the kids. Just because of my availability, it's probably going to be Jill and Jackie, but you would see both of them in one visit. And that way, if you have, because one of the questions you have to have is, what's the recurrence risk? Do we believe the mutation? Is the mutation unusual? Maybe your kid does not have KAT6A. Jill can focus on that, and then Jackie can focus more on what's the behavior? How much behind is your child in development? How can we optimize it even with clinical strategies? Things like that. So, it's a multidisciplinary clinic that has both genetics, genetic counseling, and neurological.
And I have another question. Relative to the mouse studies, given that each cat has a different truncation, do you just assume that you have a mouse and you just choose one version and generalize it?
So the way these disorders work is that it's called haploinsufficiency. You lose one copy, and because you can get a deletion and you get KAT6A deficiency. So there are a couple different ways. Sometimes the mutation can be what's called dominant negative, something that the mutation itself has a bad effect. But most of the time, in this disorder and the related disorder, it's just a loss of that extra copy. So for the mouse, you can pick something that just removes the whole gene as your first starting point. And then when you get further into it, there's now a technique where you can put patient-specific mutations. So it's often nice to do both. So for instance, Jill has created a mouse with this technology called CRISPR-Cas, where she, the knockout did not give her the phenotype that she expected. So then she went ahead and put in a patient-specific mutation, and now she sees the phenotype that she expected. So it depends a little bit on the disorder, but I think for KAT6A that just the loss of function mutation, something that knocks out the function, will give us the phenotype.
Even though I think most of the kids actually have some portion of the gene, it's just truncated.
Yeah, but the thing is, there's something called nonsense-mediated decay, and it's a system in the body that if you have a nonsense mutation that's in the middle of the protein, it just shuts down that allele. So that allele is not made into protein. There could be some kids, but most of the kids probably just only have one allele from KAT6A. Yes?
What would you advise us as a foundation in terms of what I'll call like knowledge management of KAT6A? Right, so we as a family organization, we've got a bunch of data just demographically on the patients and families. We've got their specific, every family knows their medical history and their clinical presentation of their kid. We're now getting these summary papers that are coming out describing parts of the cohort. We've got researchers, clinics like yourselves and Dr. Kelly, who are doing, I don't want to say independent research, but research locally that they'll look to contribute to the wider community. So how as a foundation or as a clinic, where should we focus all of the data together? Like how do we aggregate it all that such that there's one clearinghouse where we're pulling it together so that as new researchers gain interest and patients come online, it seems like we're still...
So NORD, and it's very important, the first step is just to figure out what's the spectrum of KAT6A. We don't even know that yet. But you can partner with NORD or Sanford, one of those places, and basically they collect all the information and then they make it available. That's the way it works. So I think that's one step. But then as you go further, there are going to be other things that you as a community want. It will be very nice to have growth charts for KAT6A, because then instead of physicians saying, oh, I think there's something wrong, your child is not growing, your child might be growing perfectly for someone with KAT6A. So that's something that down the line it would be very nice to have that. So as a community, it should, you need to create a scientific board or a medical board. Some foundations have two different ones. One is a scientific, the other one is a medical board. And then those people will interact with you and help you set your goals for that funding cycle.
Is it acceptable to have researchers on your technical or scientific board that you may want to provide some funding to, or is that a no-no?
It's better not to do that. Right. So it's best to have the researchers on your scientific board that are not actively competing for that funding cycle, and it's also good to have your scientific board have representation from many different institutions, so that people don't shun it because they're worried that there's some kind of bias in the process. So for instance, if one of us, you should only have one of us on your scientific board, and then you should have someone else from California or Europe or Asia, somewhere else. So, but there are many foundations that have already done this and done this quite well. And we're happy to put you in contact with some of the other foundations so that you can learn from them. Because there's a lot of people that have, gone through exactly the same steps.
What are the advantages of using NORD versus Sanford's way?
It's up to you. I think NORD, the function of that is to increase awareness of rare diseases. And they offer you a platform to donate money, so they help you advertise your RFA when it comes out. Sanford is more one of the other foundations that we work with and have partnered with them. They're interested in collecting, baseline data on a lot of different genetic diseases. It's free, whether it's good, I don't know. It's not what we do. But like I said, you don't have to invent the wheel. There are other foundations that have done this before you. So, yes.
Just a question back. You said theoretically you thought carnitine could work at helping the kids. Is there a natural supplement, or can you get it over the counter, or does it have to be a prescription sort of to get an extra dosage for the kids?
So we need to start, I'm going to refer that, to Dr. Kelly, but we need studies to figure out what the optimal dose. What I would like, because that's just the way I'm inclined, is I think it would be best to do those studies on the mice, because then we could look at the mouse's brain, make sure that the epigenetics are changing in the way we would predict, and that would help us rationalize to the FDA that this would be a worthwhile clinical trial. And once you've gathered those clinical data, you can start to see if there are other agents that are more potent than carnitine that could maybe have a bigger effect. Right now, I don't think we know exactly what the dose is. Dr. Kelly is kind of deciding, how to dose that. He should do that because he has a lot of expertise. But I'm just saying, as a community, it would be very helpful to replicate some of those studies in the mice, see whether we get an impact that we expect. But theoretically, carnitine is an HDAC inhibitor and should open tournament. Yes?
Thank you. Mine was related to organizing the data. Is there someone that could specifically give us the right questions to ask, for the registry to move forward efficiently so we don't hold back research?
It's an iterative process. So what's going to happen is early, there are more severe kids that are going to get diagnosed. And then over time, the kids are going to get milder, and we're going to get a bigger sense of the spectrum.
Is that based on where the mutation occurs in the beginning of the gene?
No, it could. So in some of the other disorders, the truncating mutations, the nonsense loss of function mutations, they tend to be equally severe. There are sometimes other milder mutations that are called missense, or misspellings, that sometimes are a little bit milder, but that is not, the numbers are not there yet for KAT6A.
And would having zebra models via zebrafish help? Because I know in our study there were KAT6A zebrafish.
I personally don't like zebrafish, and the reason why I don't is that the zebrafish model has undergone genomic duplication, so sometimes they have many, many copies of any given gene. So it's a little harder to interpret, and they also just, it's hard to measure their behavior. That like they swim around, but there's not really good tools to measure.
Mostly just physical.
So I personally am an advocate for doing this in mice or rats. I think that would be the best model to get preclinical. The problem is the zebrafish are really, really cheap. People can do it very, very quickly. There's also a fruit fly that people can do, but it's just very hard to study behavior. You can study other things, but it's hard to study behavior. But mice and rats, you can study behavior. Yes?
So maybe you touched on this already. I've been in and out. So when you say you're looking, for people, for grants, for things like that, who do you need and what do we tell them? Because I know most of us probably see multiple doctors in multiple clinics. I mean, we see neurology, GI, cardiology, genetics, all these people. Who do you need to do what? And how do we get them on our team? You know what I'm
Well, I
saying?
think people tend to, what I'm saying is just in general, you should advocate for your condition. So when you meet your doctors, make sure you teach them about KAT6A. They may not know, but over time, they may become advocates for this disorder. Many of the foundations have an awareness day that they do. That helps kind of raise the awareness of the condition. Some of the foundations have lobbied lawmakers in their states to increase funding for rare diseases. There are many ways to do it, and you can do it by yourself, or you can do it with something like the Nord Foundation. Oh no! Yes? Dr. Kelly, do you want to come up here and speak on the microphone?
Hi.
So you have some contact with that?
Yes, we're still it's a Duke task force for neonatal genomics. And Erica Davis was doing the functional assessment of.
Now, there are some limitations, as Dr. Bjornsson said, but we did a very interesting study with NIH on high carnitine called OPA3. And there are very specific behavioral studies of fish. And I didn't know about it before we got involved in the study. I was doing the metabolic side. And it was amazing, all these tricks they have, how they swim, how much they turn. And it's all captured on video and analyzed automatically. So if you have a decent genetic model, some questions can be asked. And we also we did the studies on the whole fish. We could take the whole fish and do the metabolic studies on the fish. So it's each system allows is very expensive, but the fish is less expensive and they each have the limitations. But I would not exclude the possibility of do have behavioral methods for studying.
We also are looking up to also share our raw data with whomever would like to have it to further research. They give us the parents and the child to share the raw data on USB, if that's helpful as well for anyone.
Okay, I think that's all the questions. I think the rest of the time we're just going to mingle